Hello everyone, welcome back to Life of Fusion, where knowledge meets excellence. So you are now in the part 5 of our lecture series, Learn from PYQs for CSIR-Life Sciences. So if you like our video, do subscribe to us and follow us and share with your friends so that everyone will get help for their CSR net preparation. So let's start the different question. So the first question. UV-induced DNA damage causes advancing replication fork to stall. So, if there is a replication is going on, okay. So this is a replication fork. Your DNA polymerase doing replication. So UV damage means UV damage means formation of thiamine dimer. So when this thiamine dimer usually what it was repaired by nucleotide excision repair, or it can't be repaired by any other repair system. Okay, so during DNA replication NER doesn't works. Okay, so here what happened, to avoid a collapse of this stalled replication for this cell uses. So what cells will do, it will do a bypass. So it's called SOS repair. In SOS repair, it's not 100% repair, it's a error prone repair. It's a error prone repair, means error will still be there. So the answer will be your lesson bypass. Non-homologous end joining is not happens during DNA replication. Mismatch repair happens during DNA replication, but if there is a replication error, okay, or here DNA polymerase is not stall. Base excision repair is happen when there is a adduct, a chemical adduct is the like your DNA analogs, okay. It feeds a chemical adduct is there in the DNA bases. Okay. So next question from genetics. What kind of inheritance is indicative in the pedigree chart shown below? So let's see. You can see generation 1, 2 and 3. The trait is available in all the generation. Okay, mean this is a dominant pedigree. Then you can see here this part. Affected individual may have unaffected children. Wow, affected are heterozygous. Their children are recessive. Okay, so that's why affected individual may have unaffected children. Is for autosomal dominant. For autosomal recessive, affected individual may have unaffected parents. That's the opposite side. So this is a autosomal dominant pedigree. So next, one more important question from genetic mapping. In Drosophila melanogaster, trait A, B and C result from recessive alleles that are located on one of the autosome. Okay, the resulting F1 females were test crossed and the following progeny were obtained. 12,000 progeny were obtained, and these are the genotype and these are the number of progeny in the F2 generations. The following conclusions were made from the above data. So the answers are like the gene order and the map distance. So here you have to find the gene order and distance between all gene. So you have to do genetic mapping. So what is the formula first here? First you have to choose what is the parental genotype. Because here they didn't provide the parental genotype. So you have to find the parental genotype. So the highest number of progeny that will show the particular genotype is called parental genotype. So you can see 350 and 380. These are two parental genotypes. So what are the parents? Parents are A plus, B plus, and C plus. Parent one more, A, B, and C. Now next step is to find the gene order. So gene order can you find from the what? Double crossover product. So how to find which is double crossover product? The lowest, what the lowest number of offspring they showed the particular genotype is known as your double crossover product. So here the 10 and 15, these two are double crossover product. So double crossover product are A, B, C plus, then A plus, B plus, C. So how to find? So the gene order here. So remember that from double crossover product, when you get, so the product will have two gene from one parent and one gene from the second parent. So that gene, the other gene is the middle gene. I'll show you here. As see here, this one, small a, small b, and C plus. A, B, and C plus. So A, B, you can see A, B is from, if A is parent 1, if A is parent 2. So A and B are from parent 2 and C plus is from parent 1. From here also you can see A plus and B plus are from parent 1 and C is from parent 2. So the other part, the third one which came from the other parent is the middle gene. So C is the middle gene. So what is the gene order here? A, B, C. Sorry, gene order will be A, C, and B. Or you can say B, C, A, whatever. So now you know the gene order. So let's write the parent name in parent genotype in the proper order. So parent 1 will be A plus, C plus, and B plus. Parent 2 will be A, C, and B. So now the next step is we have to find the single crossover product. So first we'll see if there will be a single crossover between A and C. So what will be the product? A plus, then C, then B. A plus, C, and B. See here, A plus, C, and B. So this is single crossover between A and C. Then one more you will get A, C plus, B plus. You will cross A, C plus, and B plus. Is it clear? So A, C plus, and B plus. A, C plus, and B plus. It is your single crossover between A and C. Then next two, to save your time, next two will be your double single crossover between B and C. Still we'll see here. So if A plus, C plus, B plus, A, C, B. Okay. So single crossover between B and C. Product will be A plus, C plus, and B. A plus, C plus, and B. Single crossover between B and C. Then A, C, B plus. A, C, B plus. Single crossover between B and C. So what is the formula? So the distance between A and C is number of single crossover product between these two genes. Okay, so single crossover mean 25 and 80, sorry, 25 and 35. 25 plus 35 plus number of double crossover product, because double crossover also occurred between these two genes. So 10 plus 15 divided by total number of progeny into 100. Similarly, for B and B to C, what is 100 plus 85 plus 10 plus 15 divided by total number of progeny is 1000 into 100. If you solve, you will get this at 8.5 centimorgan, and this you will get as 21 centimorgan. Okay, so let's see the option. Option A, gene order is ABC. No. Option B, ACB. Option C, BCA. These two can be ACB and BCA, as I told you. D, CBA, not. So B and C, now you got is find that B and C says that proper gene order. So distance between here is so distance between A and C. A and C is 8.5 centimorgan and B and C is 21 centimorgan. So option B and C both are correct statement. So third option is correct. Okay. So next question. In a mammal, coat color is governed by gene B. The coat color is either black or brown depending on whether the genotype is capital B or capital B small b. It means that the homozygous dominant is black color and the heterozygotes are brown color. Okay. It is not known which of these genotypes lead to the black and brown color. Okay, let's see here. It's not known. Okay, so mostly if you see here black and brown, you can say capital B black, capital B small b, you can keep it just brown. So genotype small b results in albino coat color, and further the genotype small c replace the expression of coat color resulting in albino. So small b and small c results in albino. So an albino male was crossed with a brown female, and resulting progeny has individuals with either black or brown coats. Okay, so if I say, if you don't know this black or brown, we have only know, you know that capital B and capital B small b, either of these is either black or brown. So the cross made between albino male. So the albino male should have the genotype either small b, or it also have this C gene having small c. Okay, with a brown female. So brown female, we don't know, it can be capital B or capital B small b, and C should be capital. This we remember. Okay, so from this observation, it can be inferred that the genotype of the male and female were crossed. So what is the genotype of their parents? So you see, one should be albino. It means that either small b or small c. So if you see small c here small c, here small b, here small b. Okay, then so the individuals, their progeny, what are their progeny? Their progeny are either black or brown. It means that the male is albino, but the female is not albino, and their child, none of their child receive small c or small b. So their child don't have small c, small b. Okay, so if you cross between any of these, so from where you will not find a child with small c or small b. Okay, if you cross one, capital B, small c, capital B, capital C. So there is no small c. So the chance of getting the progeny will be black or brown. So then the progeny either black or brown. Okay, so here see capital B and capital B small b. So they will get either capital B or capital B small b or capital C small c. Their genotype here. 2. What is the genotype in 2? Okay, if you see 2, 2 have a small b and here also one small b, means the chance of get the progeny will be albino, so we can cross 2. If you see 3 is that capital C, capital C, capital C, capital C. Here only capital B and small b, only heterozygous you will get. So similarly here we have only you get heterozygote, either black or brown, means both are there. That means what? Is see here don't get confused. Here that is here they says that resulting progeny had individuals with either black or brown coats. Okay, so means progeny are either black or brown, means black progenies are also there and brown progeny are also there. Okay, so if you cross these 3 and 4, from 3 and 4 you will get only capital B small b, means what? They are saying that the progeny should have capital B small b, capital B and capital C small c. So you have to find those genotype which you cross, then you will get all these 3 type of genotype. So option 1 is the correct statement. So next question, which one of the following amino acid is characteristic of and is conserved in response regulator of plant 2 component system? Plant 2 component system, if you read plant hormone, cysteine, cytokine. In plant hormone cytokine, it exerts its effect in gene via 2 component system, like bacterial 2 component system. In the 2 component system, there is a receptor domain. Okay, in the receptor there is an amino acid and one more is the regulator. Okay, so from here receptor it will induce the regulator. So here most in plant always the receptor domain is a histidine and the regulator domain is a aspartic acid. So the asked question was conserved regulator, response regulator. Which is response regulator? Aspartic acid is the response regulator. Okay, so see you in the next video. So if you like, then subscribe and share with your friends so everyone can get helps, and we'll upload some more video. If you have any doubt, you have any consultancy required for CSIR exam, feel free to contact us at our email ID which is provided in the description. Thank you everyone.